Hello and welcome. Today we're going to explore a fundamental skill in biology, preparing a temporary mount for viewing under a light microscope. By the end of this lesson, you will be able to make a temporary preparation of cellular material that is suitable for viewing with a light microscope. This is a crucial technique for observing cells and tissues in a living or near-living state, allowing us to see their structures clearly. So what exactly is a temporary mount? A temporary mount is a preparation of a specimen on a microscope slide using water or a stain that is meant for short-term observation. The key word here is temporary, nigh. Unlike permanent slides, these are designed for immediate viewing and are not preserved for long-term storage. This allows us to see cells in a more natural state. Now let's walk through the steps to create a temporary slide. We'll break it down into 6 clear stages. First, you need to obtain your specimen. Thin sections of tissues, like the epidermis of an onion or cheek cells, are ideal because light can pass through them easily. Second, carefully place a thin section of your specimen, for example, a piece of onion skin, in the very center of a clean glass slide. You can use forceps or a needle for this. Third, add a drop of water or a stain to the specimen. Water is used to keep the specimen hydrated and prevent it from drying out. Stains, which we'll discuss more in a moment, are used to highlight specific structures within the cells, making them easier to see. Fourth, add a cover slip. This is a crucial step. Gently lower a cover slip over the specimen and the liquid. The best way to do this is to start from an angle and slowly lower it down. This technique helps to push out any air and prevent air bubbles from getting trapped under the cover slip, which can obstruct your view. Fifth, remove any excess liquid. Use a piece of filter paper or a tissue to soak up any water or stain that has seeped out from under the cover slip. This keeps your microscope clean and prevents the cover slip from sliding around. Finally, you're ready to view your slide under the microscope. Always start with the low-power objective, usually 4x or 10x, to locate your specimen. Once you've found it, you can switch to a high-power objective like 40x for a more detailed observation. Let's visualize this process. This diagram shows the temporary wet mount technique. First, you place a drop of sterile water on the slide. Then, you place your specimen into that drop. Next, you carefully lower the cover slip over the specimen and the water. Finally, you observe the slide under the microscope. This visual representation helps to reinforce the steps we just discussed, showing the correct order and technique for each stage of the preparation. We mentioned using stains. But why are they so important? Many cellular structures are transparent or nearly transparent. Stains are dyes that bind to specific components within the cell, making them visible and enhancing the contrast. This allows us to differentiate between different parts of the cell. Let's look at some common stains and their purposes. Iodine solution is often used for plant cells. It stains starch granules a dark blue or black color, and it also helps to enhance the contrast of the cell wall and nucleus. Methylene blue is a common stain for animal cells, like cheek cells. It stains the nucleus a dark blue, making it very easy to identify. Acetic orcine is a specialized stain used to visualize DNA and chromosomes, especially during cell division. It stains them a reddish-purple color. So the choice of stain depends on what you want to observe. Each stain has a specific affinity for certain cellular components. Finally, a quick word on safety. When working in a lab, safety is always the top priority. First, always handle glass slides and stains carefully. Glass can break and cause cuts, and stains can irritate your skin or eyes. Always wear appropriate personal protective equipment, like gloves and safety goggles. Second, dispose of used slides and tissues properly. Never put them in the regular trash. Your teacher will provide specific instructions for disposal, which usually involves a designated biohazard container. Following these simple safety tips will ensure a safe and productive learning experience. Let's quickly recap what we've learned today. We've learned that a temporary mount is a preparation for short-term observation. We walked through the 6 steps to make one: obtaining the specimen, placing it on the slide, adding water or stain, adding the cover slip, removing excess liquid, and viewing it under the microscope. We also discussed why stains are used, to enhance contrast and highlight specific structures, and looked at examples like iodine, methylene blue, and acetic orsin. Finally, we covered essential safety tips for handling glass and stains. You are now equipped with the knowledge to make temporary preparations of cellular material suitable for viewing with a light microscope. Thank you for watching.